Hi everyone. In the last video, we zoomed in on microbiology and explored what microbes do. But for most of human history, people didn't even know microorganisms existed. And yet, humans were already using microbes to make bread, brew beer, and ferment food long before anyone could see them. So in this video, we're going to take a quick look at how our understanding of the invisible world began and how scientists eventually discovered that microbes are responsible for fermentation and disease. By the end of this module, you should be able to explain how early observations and experiments led to the discovery that these microbes drive processes like fermentation and disease. One of the oldest recorded recipes in human history wasn't for food, it was for beer. About 4,000 years ago, in ancient Mesopotamia, the Sumerians wrote a hymn to a goddess named Ninkasi, the goddess of beer. But it wasn't just a religious text, it was actually a step by step guide to brewing beer. And here's the wild part, they had no idea how it worked. They didn't know about cells. They didn't know about enzymes. They definitely didn't know about microorganisms. But they knew that if you mix grain and water and let it sit, something magical happened. The same thing was happening all over the world. In Egypt, people were baking bread that rose. In China, people were fermenting rice and alcohol. Across cultures, humans figured out how to harness these processes. Excuse me. Nearly every agricultural society developed some form of fermented food bread, beer, wine, soy sauce, pickles, etc., without ever seeing the organisms responsible. What they were unknowingly working with was a microorganism called Saccharomyces cerevisiae, at least for the bread and the beer. These are better known to you maybe as yeast. This tiny organism consumes sugars and produces carbon dioxide and alcohol. That gas makes bread rise and it produces the alcohol that makes beer. So long before we ever discovered microbes, we were already depending on them. We just didn't know that they were there. For thousands of years, people knew how to make this wine, but they didn't know why it sometimes worked and sometimes it failed. A batch could turn out perfectly or it could spoil, become sour, cloudy, undrinkable, and no one knew why. In the 1800s, the French wine industry had a serious problem. Entire batches were going bad, costing huge amounts of money. So they turned to a chemist, Louis Pasteur. Now, Pasteur didn't start with the idea of germs. He started with a simple question. What is actually happening inside these bottles? When he looked closely, he found something remarkable. Good wine contained one kind of microorganism, yeast. That's all. Spoiled wine contained multiple different kinds of microorganisms. It wasn't magic. It wasn't chance. It was life, invisible life. Pasteur showed that fermentation was a biological process, microorganisms converting sugar into alcohol. Pasteur showed that fermentation was driven by living microorganisms. But that raised a bigger question. Where were these microbes coming from in the first place? At the time, many scientists believed in something called spontaneous generation, the idea that life could arise from non living matter. Pasteur wanted to test that idea. He designed a simple but brilliant experiment. He placed nutrient broth in flasks and boiled them to kill any existing microbes. But here's the key the flasks had these long curved necks. They're called swan necks, where air could get in, but dust and microorganisms got trapped in the bin. And what happened? Nothing. Nothing at all. The broth stayed clear. No life appeared. But when that neck was broken, suddenly microbial growth exploded. Microbes weren't appearing out of nowhere, they were coming from the environment. This experiment delivered a major blow to spontaneous generation and helped establish a new idea. Life comes from life. Now, a little bit later in the late 1800s, farmers were facing a deadly problem. Livestock, especially cattle and sheep, were dying from a disease called anthrax. The disease spread in predictable ways, but no one knew what caused it. Enter Robert Koch. Koch took that what Pasteur had shown that microbes exist and come from the environment and asked a new question. Can a specific microbe cause a specific disease? He examined the blood of infected animals and consistently found the same bacterium. Then he did something bold. He isolated that microbe and introduced it into healthy animals. Those animals developed the same disease. For the first time, someone had directly linked a microorganism to a specific disease. Now, to make this idea rigorous, Koch developed a set of criteria, now known as Koch's postulates. In simple terms, they say the microbe must be found in diseased organisms, right? All diseased individuals. It must be able to be isolated and grown, and it must cause the disease in a healthy host. It also must be able to be re-isolated from that host once it's sick. Um, and this kind of gave us this systematic way of identifying disease-causing organisms. And with that, germ theory was born. So for thousands of years, humans were using microbes, baking bread, brewing beer, making wine, without ever knowing they existed. Pasteur showed that these processes weren't magic. They were driven by living, invisible organisms. He proved those organisms came from the environment. And Koch took it one step further, showing that specific microbes could cause specific diseases. In just a few decades, we went from mystery to a scientific understanding of the invisible world. So, with that, I hope you enjoyed the lecture, and I'll see you in our next lecture where I'll discuss how microbes or what different microbes are.